Okay, here we go. Kinesiology 230, chapter 3, still going on with bioenergetics, and we're in presentation number 7. Now we're kind of shifting our focus over to the oxidative system and also a little bit more about the Krebs cycle. It's the primary source of ATP at rest when we talk about the oxidative system. And I gave the example of, and the irony of, the person sitting on the couch watching the sprinter on TV is actually on a percent basis utilizing fat stores in the oxidative system to a greater degree than the person who's sprinting 100 meters. Now, that's with the caveat and the understanding that the person who's sprinting 100 meters is actually working at a much, much higher calorie per hour burn rate than the person sitting on the couch, and total calories burned relative to fat might be darn equal, to be quite honest. But just realize on a percent basis, the person sitting on the couch, their percent burn of fat is much higher than the sprinter that they watch on TV. Because what happens is, when you're at rest, you're about 70-30, but when you go ahead and start with high intensity aerobic activity, it shifts the demand to almost 100% carbohydrate. And that shift, then, what ends up happening when you have prolonged activity, it kind of swings the pendulum back to more fats and proteins as an energy source. But I have to say that the use of protein as an energy source would require a very long duration submaximal cardiovascular activity. And we talked about this before in terms of glycolysis, and we talked about slow and fast, meaning aerobic versus anaerobic glycolysis, and the two main paths. And we came to the conclusion of this. And when you first look at this chart, it could almost be interpreted as right side, left side, or red side, yellow side in this rendering. But really what it comes down to is not that. It comes down to this. It comes down to how the pyruvate, that when you get all the way through the system from the blood glucose or the muscle glycogen, flows through the system, and you get through all of this, you end up with pyruvate or pyruvate. And if there's oxygen involved, it goes into the Krebs cycle. If there is not oxygen involved, it becomes blood lactate, and that blood lactate is then shipped out, sent to the liver, and that's sent to the liver for processing through the Cori cycle. And the process itself goes and basically takes the pyruvate, creates pyruvate, converts it to lactate. Lactate gets converted to glycogen. Glycogen back into the muscle, gets used in the muscle, et cetera, et cetera, et cetera, until such time that there's enough oxygen involved and that pyruvate no longer is sent out to the liver anymore. It's actually sent into the Krebs cycle in the actual muscle itself, in the mitochondria, to be able to be processed. All right, so with this, you have this side by side here. And the determination that you have, it comes down to two things like I've talked about a couple times already. It's like, is there oxygen present, and what's the level of intensity of the exercise? And really the level of the intensity of the exercise is going to dictate if there's oxygen present or not. A little bit can be a little bit misleading because, let's face it, somebody who is in better shape, who's fitter and more capable, that individual might be able to process oxygen better, like we talked about. They might have the ability to have oxygen load into the blood a little bit better, into the hemoglobin, than others. They'll have a greater affinity, like we talked about before. So it kind of depends upon the person, and it also depends upon the exercise itself. Now, we know lactate itself is going to end up going into the Krebs cycle or the liver. And if it's anaerobic without oxygen, you'll need acetyl coenzyme A to make this all happen. But it ends up going into the Krebs cycle itself. And the one thing I don't like about this rendering when we talk about this, and this is really what they're talking about, they're taking this Krebs cycle down here with oxygen, and this is what it looks like. So they have kind of a blown up picture of the Krebs cycle and showing the intricacies of the cycle. A lot of NADH, a lot of FADH, a lot of GTP, a lot of GDP. Oh my goodness. So there's a lot going on in here that it's a lot to process. And the big thing you really just want to understand is you have fatty acids, you have carbohydrates, amino acids. Those are the main things in terms of blood glucose that actually go into the Krebs cycle. So metabolism of blood glucose and glycogen begins with glycolysis. And with glycolysis, and you have O2 present, the end product of glycolysis, as we talked about, is pyruvate, and it's transported into the mitochondria in the cells where the process that's called Krebs cycle actually happens. If there wasn't O2 present, or if the exercise was too intense, I'll give you a moment, what would happen? Cori cycle. That's right, it goes into the liver. You got it. So with that, it's the pyruvate itself does not get converted to lactate. If there wasn't oxygen, it would be converted to lactate, and that would cause the Cori cycle to happen. We're talking about oxygen being present, and the pyruvate goes or is transported to the mitochondria in the cells, and that infers right there, if you see a test question somewhere else, if there's pyruvate involved, it's the Krebs cycle. If it's the Krebs cycle or pyruvate, it is oxygen related. If it is not pyruvate and it's lactate, or the liver's involved, or the Cori cycle's involved, it's anaerobic without oxygen. Start to kind of compartmentalize those and put them side by side, and you'll get through this chapter. So the pyruvate is converted to acetyl coenzyme A. That's the acetyl-CoA, and that's what enters the Krebs cycle. So what you see here is amino acids to pyruvate, pyruvate to acetyl-CoA, fatty acids to acetyl-CoA. That goes into the Krebs cycle, goes through this process, comes on out, and it ends up creating ATP. What you have to look at is the two main types of oxidation that are represented here, one of which is protein oxidation, which is a breakdown of amino acids. That's this up here. And then you have beta oxidation, which is a breakdown of fatty acids. And beta oxidation, I remember that as a student in undergrad, just thinking in terms of how you're chopping up a real complex fat molecule to make it more tolerable and usable, if you will, for the Krebs cycle to eat it. Sounds stupid, I know, but that's just how I remembered it. Anyways, beta oxidation was a process of cutting up the fatty acids. With the electron transport chain, I've talked a lot about when you look at the different etymology of words. And you have this electron transport chain, and the ETC, or electron transport chain, uses NADH and FADH2 molecules to rephosphorate ADP to ATP, meaning going anabolic, right? We're going to take ADP and make it ATP. We're going to create something, put our building blocks together to give us adenosine triphosphate. And if you want to just, again, do great on Scrabble, you have nicotinamide adenine dinucleotide for NADH, and FADH is flavin adenine dinucleotide. So it comes down to what these components are. In some regards, I can tell you in terms of a test in the quiz material, you don't have to stress too much about knowing the differences of these. You just have to understand what they do. And the hydrogen atoms themselves are passed down this chain, and they're passed down by these electron carriers called cytochromes. And the electrons, they go through the electron transport chain, and when they do this, energy is released. And this is used to put, as the book states, used to pump protons across the membrane, causing an electrochemical gradient. And this gradient helps create ATP. So the math on this is 1 NADH, that's the nicotinamide adenine dinucleotide. 1 NADH gives you 3 ATP. And then the 1 FADH, which is flavin adenine dinucleotide, gives you 2 ATP molecules. And I guess there's a couple different ways if you want to try to tackle remembering that. I don't know how you would go about doing that. I know, like, when I had to remember this, I just memorized it. I couldn't find like a mnemonic or a way to really help me out here. The only thing I could really come up with is F comes before N and 2 comes before 3. That was about the only real way I could remember this when I was an undergrad. So, with that, guys, just understand that when we start talking about processes in the body, the different components in the body, and you start to dissect the the nomenclature and the verbiage and how words are described. It sometimes helps just to kind of take a word and break it out bit by bit by bit, and that may help your cause. All right, guys, I'm going to move on to the next presentation.